There is a question that has haunted physicists and neuroscientists for over a century, and it goes something like this. You are made of atoms. Atoms obey the laws of quantum mechanics. So why does it feel like something to be you? Why does the universe, built from particles that exist in superpositions of multiple states simultaneously, produce a brain that experiences a single, continuous, vivid stream of awareness? That is not a poetic question. That is a hard physics problem, and it may be the hardest one we have ever faced. We tend to think of the brain as a biological computer, a sophisticated information processor running on electrochemical signals, and on one level, that description is accurate enough. Neurons fire, signals propagate, memories form, decisions emerge. Classical neuroscience has mapped this machinery with extraordinary precision, and it has given us everything from antidepressants to deep brain stimulation. But here is the problem. Classical neuroscience, for all its power, cannot tell you why any of this feels like anything at all. It can tell you which neurons activate when you see the color red, but it cannot tell you why there is a subjective experience of redness happening at all. That gap between the physical process and the felt experience is what the philosopher David Chalmers famously called the hard problem of consciousness, and it remains completely open. Now, some neuroscientists are comfortable sitting with that gap. They believe that consciousness is simply what a sufficiently complex information processing system looks like from the inside, and that one day we will close the explanatory gap through better neuroscience alone. But a growing number of physicists and biologists are asking a more radical question. What if the gap exists precisely because we have been applying the wrong branch of physics? What if consciousness is not just a product of classical computation happening in neurons, but is instead tied to the quantum mechanical substrate from which all matter is ultimately constructed? To understand why that is even a coherent proposal, we have to go back to the foundational strangeness of quantum mechanics itself. At the quantum scale, the rules of reality are categorically different from anything we encounter in daily life. A quantum particle, say an electron, does not have a definite position until it is measured. Before measurement, it exists in what physicists call a superposition, a mathematical description that says the particle is simultaneously in multiple states at once. This is not a statement about our ignorance of where the particle is. It is a statement about the fundamental nature of reality at that scale. The particle genuinely does not have a single definite location. It has a wave function, a probability amplitude spread across space, encoding all the possible states the particle could be found in. The double slit experiment makes this viscerally concrete. You fire electrons, one at a time, at a barrier with two slits. On the other side is a detector screen. If electrons were classical particles, like tiny bullets, you would expect them to pass through one slit or the other and build up two bands on the screen. What you actually get is an interference pattern, a series of alternating bright and dark bands that is the unmistakable signature of a wave. The electron, traveling alone, interferes with itself. It goes through both slits simultaneously. Its wave function propagates through both openings, and the two wavefronts interfere with each other, constructively in some regions and destructively in others, producing that striped pattern. Now here is where the strangeness deepens. The moment you place a detector at the slits to find out which slit the electron actually goes through, the interference pattern vanishes. You get two classical bands as if the electron were a particle all along. The act of measurement, the act of gaining information about the electron's path, collapses the wave function. The particle stops being a spread-out probability cloud and becomes localized, a definite thing in a definite place. This is the measurement problem. The very act of observation, the interaction between a quantum system and a measuring apparatus, forces what was a superposition of possibilities to crystallize into a single outcome. The universe at its deepest level seems to resist having definite properties until something interacts with it and forces it to commit. That observation collapses the wave function, is experimentally unambiguous. But what exactly counts as an observation and what is physically happening during collapse remain among the deepest unsolved problems in foundational physics. And this is precisely the opening through which the connection between quantum mechanics and consciousness enters the discussion. One of the earliest and most serious proposals along these lines came from the physicist John von Neumann in the 1930s. Von Neumann formalized quantum mechanics with mathematical rigor that has never been surpassed, and at the end of his analysis, he arrived at an uncomfortable conclusion. If you trace the chain of interactions during a quantum measurement, every component of the measuring apparatus, the detector, the amplifier, the readout device, is itself made of quantum particles and could in principle be described by a quantum wave function. The collapse of the wave function never seems to happen within the physical chain of events. Von Neumann suggested that the chain only terminates, only produces a definite outcome when it reaches a conscious observer. The mind, he proposed, is what ultimately collapses the wave function. Now, most physicists today do not hold that strong a position. The decoherence approach to quantum mechanics has shown convincingly that quantum superpositions are not fragile creatures waiting for a mind to collapse them. They are fragile for a much more mundane reason. Any interaction with the surrounding environment, any stray photon, any thermal fluctuation, any neighboring molecule, entangles the quantum system with its environment and rapidly disperses the phase coherence that makes superposition possible. This process, called decoherence, happens at an almost incomprehensible speed at the scale of warm, wet, dense biological tissue. At room temperature, in the molecular chaos inside a neuron, quantum superpositions are typically expected to decohere in timescales on the order of 10 to the minus 13 seconds. That is a tenth of a trillionth of a second. The timescales on which neurons fire and compute are on the order of milliseconds, roughly 10 billion times slower. Classical neuroscientists look at that number and say the case is closed. Quantum effects simply cannot survive long enough inside a living brain to play any role in cognition. This is the dominant view, and it deserves to be taken seriously. But nature has a habit of refusing to stay within the lines we draw for it. In 2007, a team of researchers studying photosynthesis in certain species of algae made a discovery that sent a genuine shockwave through the scientific community. They found that the process by which light energy is transferred from antenna molecules to the reaction center in photosynthetic complexes, a process that occurs with near-perfect efficiency of almost 100%, appeared to exploit quantum coherence. The energy was not hopping randomly from molecule to molecule like a classical particle. Instead, it seemed to be exploring multiple pathways simultaneously in quantum superposition, allowing it to find the most efficient route. In warm, wet, biochemically noisy plant tissue, not in a laboratory cooled to fractions of a degree above absolute zero, but inside living cells at room temperature. This was a profound shock because the conventional wisdom was that biological systems were simply too warm and too wet to sustain quantum coherence. The thermal noise of the environment should destroy any delicate quantum state almost instantly. And yet here was photosynthesis, one of the most fundamental processes in the biosphere, apparently exploiting precisely that kind of delicate quantum coherence to operate with an efficiency that no classical mechanism could explain. And photosynthesis is not an isolated case. Cryptochrome proteins in the eyes of migratory birds appear to use quantum entanglement to detect the direction of the Earth's magnetic field. The mechanism is called the radical pair mechanism. When light hits cryptochrome, it generates a pair of electrons that are quantum entangled with each other. The subsequent evolution of that entangled pair is sensitive to magnetic field orientation, allowing the bird to essentially sense magnetic north through the quantum mechanical behavior of electrons in its visual system. Birds are navigating continents using entangled electrons. The quantum house of cards does not always collapse in the biological hurricane. Sometimes biology has learned, through billions of years of evolution, to stabilize and exploit exactly the quantum effects that we assumed were too fragile to survive. This is the field of quantum biology, and it has fundamentally changed the conversation about whether quantum mechanics can play a functional role in living systems. Now, given this context, the question of whether quantum effects matter in the brain becomes considerably less easy to dismiss. The most developed and most seriously discussed proposal connecting quantum mechanics to consciousness is the orchestrated objective reduction theory, developed jointly by the physicist Roger Penrose and the anesthesiologist Stuart Hameroff. The theory is two-part, and the parts come from very different directions. Penrose's contribution comes from his analysis of mathematical computation and the nature of mathematical insight. Penrose argued, in his books The Emperor's New Mind and Shadows of the Mind, that human mathematical understanding cannot be reduced to any algorithmic computation. His argument is based on Gödel's incompleteness theorems, which demonstrated that within any sufficiently powerful formal system, there are true statements that cannot be proven within that system. Penrose claimed that human mathematicians can, in principle, see the truth of such statements in a way that no fixed algorithm could. If that is correct, then whatever the brain is doing when it performs mathematical reasoning, it is not performing a classical algorithm. It is doing something that transcends Turing computation. And Penrose argued that the only physical process we know of that is not reducible to classical computation is the collapse of the quantum wave function. This is a deeply controversial claim, and many mathematicians and philosophers reject Penrose's reading of Gödel. But even those who reject it acknowledge the force of the underlying question. Is the brain fundamentally a classical computer, or is it doing something else? Hameroff's contribution comes from the biology. He pointed to a specific cellular structure called microtubules as the possible site of quantum computation in neurons. Microtubules are cylindrical protein polymers that form part of the cytoskeleton, the internal scaffolding of cells. Every neuron is filled with them. They are composed of tubulin protein subunits, and each tubulin can exist in different conformational states, essentially different shapes, depending on the distribution of electrons within the protein. Hameroff proposed that these conformational states could exist in quantum superposition, making each tubulin a biological qubit, and the highly ordered periodic structure of the microtubule lattice might provide the kind of shielded, coherent environment that protects quantum states from thermal decoherence longer than we would naively expect. Together, Penrose and Hameroff proposed that microtubules maintain quantum superpositions of tubulin states, process quantum information, and then undergo objective reduction, a hypothetical physical process that Penrose derived from an attempt to reconcile quantum mechanics with general relativity. In general relativity, space-time geometry is determined by mass-energy distribution. A quantum superposition of two different mass distributions would correspond to two slightly different space-time geometries existing simultaneously. Penrose argued that this is physically unstable. Nature cannot sustain two different geometries of space-time indefinitely. At some point, the superposition must resolve, and Penrose proposed that this resolution happens not through environmental decoherence, but through a fundamental feature of the universe's gravitational geometry. This is the objective reduction. It is not triggered by an external observation. It is triggered by the internal quantum gravitational instability of the superposition itself. The orchestration part comes from Hameroff's claim that the timing and pattern of these collapses in microtubules are orchestrated by biological processes, synaptic inputs, and cellular metabolism. And the moments of objective reduction, these events where the quantum wave function resolves due to space-time curvature, are the moments of conscious experience. Each collapse is, on this theory, a fundamental act of choosing, a moment where the quantum possibilities of tubulin states snap into definite configurations, and that snapping is the physical correlate of a moment of awareness. It is important to be clear about the scientific status of this proposal. OR is speculative. The experimental evidence for quantum coherence in microtubules, specifically functioning on the timescales relevant to neural computation, has not been established. Critics, including the Nobel laureate Max Tegmark, have published detailed calculations suggesting that the decoherence timescales for microtubules in the brain are far too short, by many orders of magnitude, for the mechanism to work. Hameroff and Penrose have responded with counterarguments, suggesting that the ordered water molecules surrounding microtubules and the protein lattice structure itself may provide a more shielded environment than Tegmark's calculation assumed. The debate is ongoing, and it is a real scientific debate, conducted in peer-reviewed journals with competing calculations and experimental programs. What matters from a physics standpoint is that the question is being asked rigorously and that the experimental tools to begin probing it are becoming available. Advances in quantum sensing technology, in ultrafast spectroscopy, and in cryo-electron microscopy are giving researchers the ability to look for signatures of quantum coherence in biological systems at a resolution and timescale that simply did not exist a decade ago. The question of whether microtubules maintain biologically relevant quantum states is, at least in principle, an answerable question. But let us step back from the specific machinery of OR and think about the deeper physical picture that the quantum biology revolution has forced us to confront. For most of the 20th century, the dominant assumption in both physics and biology was a clean conceptual division of labor. Quantum mechanics rules the atomic and subatomic scale. Classical physics handles everything above it. Life, brains, cognition, all of that happens at scales where classical rules apply. The two worlds were assumed to be cleanly separated, with decoherence acting as the wall between them. What quantum biology has shown us is that this wall is not impermeable. Evolution has found ways to use quantum effects at biological scales to harness entanglement and coherence as functional tools in the chemistry of life. The boundary between the quantum world and the classical world is not drawn at the edge of a molecule or the edge of a cell. It is dynamic, context-dependent, and in some cases actively managed by biological machinery that has been shaped by billions of years of natural selection. If that is true for photosynthesis and for bird navigation, it forces us to revisit the assumption that the brain, arguably the most computationally sophisticated biological structure we know of, has no access to those quantum resources. It does not prove that the brain uses quantum computation. It does not validate OR, but it removes the comfort of the simple dismissal. We cannot just say the brain is too warm and wet. We have to actually look. And looking at the brain as a potential quantum system opens up a genuinely new way of thinking about what computation and information processing might mean in biology. A classical computer processes information through definite bit states, zeros and ones, and the rules that govern their transformation are deterministic at the level of the hardware. A quantum computer processes information through qubits that can be in superpositions, and the transformations are governed by the continuous evolution of wave functions before a final measurement extracts a classical outcome. The computational power difference is not just quantitative. For certain classes of problems, quantum algorithms provide an exponential speedup that is simply not achievable by any classical algorithm, regardless of the hardware. If neurons or the molecular structures within them are performing something analogous to quantum computation, the implications for our understanding of cognition are not trivial. It would mean that the speed of certain perceptual and cognitive processes, the apparent simultaneity with which the brain integrates information from multiple sensory streams into a unified conscious experience, might reflect quantum parallel processing rather than an extraordinarily rapid classical serial computation. It would also potentially offer a physical mechanism for the kind of non-local integration across distant brain regions that is one of the most puzzling features of conscious experience under classical models. None of this is settled science. All of it is at the frontier, where the instrumentation is just barely reaching the required sensitivity, where the theoretical frameworks are being constructed and contested in real time. This is science in the making, not science in the textbook, and that distinction matters. But here is what we can say with confidence. The universe, at its most fundamental level, is a quantum mechanical universe. Every atom in your brain, every electron threading through your neural tissue, every protein folding and unfolding in your synapses obeys quantum mechanical rules. The wave function is not a feature of exotic laboratory physics. It is the fundamental description of every particle of matter everywhere. The question is not whether the quantum world is relevant to the brain in principle. It is whether the quantum world is relevant to the brain in a functionally meaningful way at the scale of neural computation and conscious experience. That is a question about where the boundary between quantum and classical behavior falls inside the specific physical environment of biological neural tissue. And we do not yet know the answer. What we do know is that the boundary is not where we thought it was. Quantum biology has moved it. The brain remains the last and most important frontier. The physicist's instinct, developed over a century of wrestling with quantum mechanics, is to resist the temptation to draw clean lines. Every time we have said, quantum effects stop here, classical physics takes over here, reality has found a way to blur that line. The electron behaves as a wave until it does not. The photon travels all paths until it arrives. The tubulen sits in every state until it must choose one. Perhaps the mind, the one thing in the universe that observes and collapses and makes real, is itself built from the very machinery that does the collapsing. Not as a mystical proposition, but as a straightforward consequence of the fact that observers are made of quantum matter too. The wave function that governs the universe does not stop at the skull, it runs all the way through.